All right, so now we're going to talk about making the red blood cells. This basically, we're going to go over this picture here, and it's an 8-stage process, and yeah, you should pay attention to it. All right, so that's what we're going over. You'll see it again at the end. So erythropoiesis is the making of the red blood cells, and that will take 15 days or so. And like I said, 8 steps. The hematopoietic stem cells will become what we call myeloid cells, and the myeloid stem cells will then modify again to become pro-urethroplasts. Pro meaning before. So erythrocyte is a mature red blood cell. If you see blast, that's going to be an immature. Anytime you see blast, it means the baby form of the cell, or you think it was the baby form of the cell. If you're cyte, think of the mature form of the cell, cyto for cell. So erythroblasts will be the baby form. Pro is what we have even before that. Sorry, just breaking down the name to make it easier to remember. So the pro-urethroblast will divide many times, and it will make a bunch of basophilic erythroblasts. Basophilic because they like the basic dye, that purple dye, whenever we're doing a Wright's blood stain. And that leaves like a purplish blue color. These things have lots of ribosomes. That's one of the ways you know they are not a mature red blood cell. A mature red blood cell doesn't have ribosomes anymore. All right, so polychromatic erythroblasts, poly for many and chrome for color, okay? They make a lot of red colored hemoglobin, and so we now have the blue staining from the basophilic and the red from the hemoglobin, so there's more than one color. That's where the polychromatic comes from. Ortho means all the same, okay? So orthochromatic, you basically have hemoglobin. You've gotten rid of most of the organelles. You're starting to, the nucleus is breaking down and you're spitting out other things. Getting rid of the nucleus is why the sides go and give you that concave in the middle. And boom, the next step after the orthochromatic erythroblasts would be to call them reticulocytes. Those are almost, a reticulocyte is almost to the stage of being a grown-up red blood cell. So that's the 7th stage. And the only difference is they still have a little bit of ribosome. Once they've gotten rid of the ribosomes, then we're going to say they're a mature erythroblast. Mature erythrocyte. And that'll happen in 2 days or so. The ribosomes degrade and it's ready to go. The reticulocyte count, that's one of the things you'll see in diagnostic tests is the reticulocyte count. What they're doing is counting the number of cells that are right on the edge of becoming functional red blood cells. So, second. Okay, so here's a picture of that process. You've seen this picture before. This is erythropoiesis, the making of the red blood cells. Next, we're going to talk about how you control that. So, if you get too few red blood cells, if the blood doesn't have enough of them, you're going to have trouble getting oxygen to the tissues. The tissues will have hypoxia. Fancy clinical name for not enough oxygen. Too many red blood cells, the blood will get too thick. We call that polycythemia. We'll probably be mentioning that in a little bit when we talk about hormonal control and the abuse of EPO, erythropoietin. EPO or EPO is the chemical that helps tells red blood cells to mature. Anyway, red blood cells don't last very long, like 100 to 120 days, because they can't repair themselves. So we are making new red blood cells all the time. We're making about 2 million red blood cells every second, because that many of them are dying. They're mostly getting cleaned out in the spleen whenever they get old and stiff and lose their flexibility. They get caught in the spleen trying to wiggle through all the maze of twisty little passages all alike in the capillaries, and they, yes, I get points for a video game reference. And they then are removed there, and we recycle the heme because metal is precious. Anyway, the balance, we have to do a balance between making red blood cells and destroying red blood cells. That's going to depend partially on what hormones are telling us to do and partially on what we could do with the nutrients that we have in our body from our diet. So dietary requirements. All right, so first, hormonal control. We're talking about erythropoietin, or EPO. This is a hormone anything you hear in this chapter about EPO, you're going to hear again when we do endocrine, because this is an important one. EPO stimulates the formation of red blood cells. You always have a little bit of EPO in your body, in the blood, to maintain some level of making red blood cells. It's given off by your kidneys, a little bit from the liver, but mostly the kidneys. And this happens whenever you are at low oxygen. When you're having hypoxia, the kidneys start screaming, hey, 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 we need oxygen. And the way they do that is releasing EPO into the body. So at low oxygen levels, oxygen-sensitive enzymes in the kidneys can't degrade hypoxia-inducible factor, and the HIF accumulates, which triggers the release of EPO. And that EPO is going to say, hey, red blood cells, we need red blood cells, make them mature faster. So what can cause this? Hypoxia, that could be caused by a decreased number of red blood cells. Maybe you had a hemorrhage. Maybe you've been destroying more of them than normal. Maybe you have the normal number of red blood cells, but you're having trouble putting hemoglobin in them, maybe because you have an iron deficiency. Maybe you have reduced oxygen levels available. You're at high altitude, or you're having lung problems like pneumonia. Some people do this on purpose. We're about to start the Olympics. This is why Olympic athletes in the United States train in Boulder, Colorado, because it's very high altitude. The air is very thin. The air has less oxygen. And so you will naturally have your kidneys say, make more red blood cells, make more red blood cells. An athlete who trains up in Boulder is naturally going to have more hemoglobin, higher hematocrit in their body than someone who trains, say, in Miami, right on the coast. Now, other athletes in other countries, they will simulate this in other ways. There are some sports where people are from countries with low altitude. They will actually have them sleep at night in a bubble, a plastic bubble that has lower than normal levels of oxygen. I know that sounds weird, and it is, but the practical upshot is that they will act like they're at very high altitude. Their body will make more hemoglobin, more red blood cells to compensate, just like they were training in the mountains. Anyway, but we'll talk about that kind of stuff later. So if you have too many erythrocytes or high oxygen levels, that's going to inhibit EPO production. Remember, I said before, too many erythrocytes, the blood could actually get sticky and thick and hard to pump. If that condition happens, we call it polycythemia. Poly for many, cyto for cell. Anyway, you don't want to have polycythemia. So if you have if the blood's starting to get thick, the kidneys are going to cut down on EPO production. What EPO does is make the red blood cells mature faster. We've already said that. Just, you can see here it is on the slide now. Testosterone will enhance EPO production, resulting in higher red blood cell counts in males. If you remember, when we look back here, going back to the hematocrit, right there where we remember hematocrit is the level of red blood cells that we have, and you can see that, why is it not right there? The numbers, let's see, so there we go. All right, so for males, 47% hematocrit would be normal, for plus or minus 5. For females, 42 plus or minus 5. That difference is caused by what we just read about. That is caused by the fact that testosterone ramps up EPO production. So, all right, back here. There we go. All right, so here we have a feedback mechanism that they're showing. Stimulus, hypoxia, that's going to cause the kidneys to produce HIF, which is going to cause EPO to be released, and that will cause the red blood cells to mature, which will cause there to be more red blood cells, which will increase the oxygen level, which will get rid of the stimulus, and so then you'll lower the EPO production. Anyway, they're going step by step through these pictures, but you got it. All right, so now we talk about the abuse of EPO. We're talking blood doping here. We're talking something that used to be a huge deal in cycling. So the use of EPO increases their hematocrit, allows the athletes to increase stamina and performance. It's dangerous because EPO could increase the hematocrit levels from the normal 45%, 45-ish% up to maybe 65%. And then when the athlete sweats and they lose water from their body, that can get even more concentrated. That's how you get your polycythemia. The blood gets thick like sludge, and then it clots. You can have strokes. This is something that back in the 70s, they had a problem with a lot of really healthy people having heart attacks during sporting events, dying. Most of those people were doing blood doping in one form or another. And one method that could be used, the method Lance Armstrong got busted for, is EPO. Actually, that's not true. One of the methods people use is taking EPO. What Lance did, what Armstrong did, was take an EPO precursor. They had started tests for EPO, and so what he did, they took a chemical that will turn into EPO. He's like, oh no, I'm not taking EPO, because he wasn't. It still was giving him an edge, so it still was cheating. All right, so what do we need to make red blood cells, to do erythropoiesis? We talked about the hormonal control, now let's talk about the diet. You need amino acids because hemoglobin is a protein, and proteins are made out of amino acids. You need lipids because we're going to make phospholipids. We're going to make membranes for the plasma membrane of the cell. And you need to make carbohydrates. You need to have carbohydrates to power the cell because remember, we're doing glycolysis to do the anaerobic respiration to power the cell. So you got to have iron. 65% of all the iron in the body is in the hemoglobin. The rest of it's in the liver, the spleen, the bone marrow, places that either remove hemoglobin or make hemoglobin. So, yeah. Free iron ions are super toxic, so we always have the iron connected to something else, so it's not free. And in cells, you can store it as ferritin or hemosiderin. You can transfer it through the blood by binding it to a protein called transferrin. That's the kind of protein I like, a name that tells you what it does. Anyway, You need vitamin B, you need folic acid. If you can't do those, you can't make DNA. And yes, I know that the red blood cell doesn't have any DNA in its grown-up form, but as a baby, it totally does. Remember, going through the stages, we're getting up to the reticulocyte, we're shedding mitochondria. Uh, all of that stuff, all the organelles we need beforehand, all the making things, that has DNA, and that means we need B12. All right. So these red blood cells last 100 to 120 days, and they are anucleate. Fancy way of saying they got no nucleus. You already know that. We already talked about that. They shed it as they grow up. That means they can't make new proteins. That means they can't grow, they can't divide, and they can't repair themselves. So old red blood cells, as they get older, they get really fragile. They break easily. The hemoglobin begins to break down. It says they can get trapped in small circulatory channels, especially in the spleen. Like I said before, maze twisty passages all alike. That's the capillaries. Macrophages in the spleen engulf and break down dying red blood cells. Okay, so what is a macrophage? We already talked about it, but we never used that name. A macrophage is a grown-up monocyte. Remember, never let monkeys eat bananas, and monocyte was one of the white blood cell types? Well, a macrophage is a grown-up form of that. And the name should tell you what it does. Macro means big, phagy is to eat. This is the big eater. It's like a big Pac-Man comes on, wah, 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 eating things. And the ones in the spleen eat and break down to old dying red blood cells. Why? So we can keep the iron and we can use it again. Okay? Well, actually, that's not true. They break them down so they don't clog up capillaries and cut off oxygen. But what do we do with it when we break them down? We keep the valuable parts we can't replace easily. Moving on. All right, so as we break down the red blood cell, the heme, the iron, and the globin are separated. The iron binds to ferritin, hemoceritin, and we're going to store it to use it later. That's how we store it in the cells. The heme is broken down into bilirubin, and the liver is going to take the bilirubin and it says it secretes it into the intestines. That's true. It does that as part of the bile. We hear people talk about gallbladder. The gallbladder is full of bile. Well, a large part of that is the broken down heme groups from the red blood cells that broke down. And that bilirubin will break down into urobilinogen. That urobilinogen is then turned into a brown pigment in the liver called stercobilin, and that is the stuff that is in the bile that goes into the intestines, and that turns the poop brown. Okay, why? It's full of iron. Or sorry. Why? It's yellow stuff. It's red stuff that turns to yellow stuff that turns to brown stuff that then dyes the feces. What I said before a moment ago. Dang, that C is fast. What I said before is false. We're not trying to get rid of the iron. Some of that may happen inadvertently, but iron precious, we keep that in the body so we can make more red blood cells. The globin, the globular protein, we break that down into the individual amino acids and release those into circulation. They go off to where they can be used again to make other proteins or the same protein again. All right, moving on. There is a pretty picture with what's happening in each stage of the life cycle. We've been over that in the text, and they've got the same picture labeled 7 times. Blah. Okay, erythrocyte disorders. Most of the problems with red blood cells, erythrocytes, are either anemias or polycythemias. Anemia is when the blood is low in oxygen. It's not carrying much oxygen, and you don't have good metabolism because of that, you have a problem. Usually this is a sign of a problem, not the disease itself. What we're saying is this is a symptom. I wish I just said symptom instead of sign of the problem. So what does anemia cause? Fatigue, pallor, dyspnea, chills, and there are three groups based on cause. You can have anemia from blood loss. You just run out of blood. You don't have enough blood. Probably due to, say, a hemorrhage or a cut where you're bleeding out. You could have anemia because you're not making enough red blood cells, or you could have anemia because you're killing too many red blood cells. So pretty straightforward. Blood loss. We already said hemorrhagic anemia, rapid blood loss from a wound. You treat the wound to close it so you're not bleeding out. That's easier if it's external hemorrhage and internal hemorrhage. Those are hard to treat. You can have chronic hemorrhagic anemia. Chronic means it goes on and on and on. This is a long-term condition. Hemorrhagic is the process of bleeding out. And anemia, of course, that's what we're talking about, having low oxygen getting to the cells. So a slight blood loss that keeps going on and on and on, that's chronic hemorrhagic anemia. That could be caused by a bleeding ulcer, it could be caused by hemorrhoids, which common parlance, common slang people call it piles. The primary problem, the thing you have to do, you got to stop the bleeding. Plain, straight and simple. All right, so you could also, so those were anemia from bleeding too much. Now we're going to talk about anemia from not making enough red blood cells. And we'll go into more detail on this, but this could be caused by not having enough iron, so you can't make the heme groups. It could be pernicious anemia, that's an autoimmune disorder. It could be renal anemia, where your kidneys are not making the EPO to tell you to make or mature the red blood cells. Well, let's go through each one of those. Iron deficiency anemia could be from hemorrhage, but also from not getting enough iron, not eating enough, or you're not absorbing enough. It's possible to have conditions that cause you to eat, you consume enough iron, but you can't use it, you can't absorb it. You're going to make red blood cells that are called microcytes. Micro for small, cyto for cell. These are little tiny cells. They're not as big as a normal red blood cell. They're very pale in color because they don't have any hemoglobin or have very little hemoglobin. You can't make the hemoglobin because you don't have enough iron. What do? Probably give the person iron supplements. All right, you could also have pernicious anemia. Remember, anemia, general term, you don't have enough functional red blood cells. So pernicious, you could have an autoimmune problem. There's a thing that the stomach makes in digestion. It's called intrinsic factor. This is one of the problems when people have like a stomach bypass or something. If you remove that stomach, you got to remember to take the intrinsic factor that the stomach would be making. Stomach doesn't just do digestion, it does other things. It makes intrinsic factor that lets you absorb the B vitamins. Remember, you got no B vitamins, you got no DNA reproduction, which means you don't have a lot of cell division going on. Anyway, sorry, I should have just read, that's what it says. Autoimmune disease that destroys stomach because it produces intrinsic factor. Intrinsic factor needed to absorb B12. B12 needed to help red blood cells divide. Without the B12, red blood cells will enlarge and get bigger, but they can't divide, resulting in microcytes. You can treat this by giving people B12 injections, or, I know this sounds odd, giving them B12 nasally as a nasal gel. This can be caused by low dietary intake of B12, but we just gave you some treatments. And it can be a problem for vegetarians. They have trouble getting enough B12. They also can have trouble getting enough iron. But take your supplements, balance your veggies carefully. Don't just eat random veggies. Same thing. The globins are going to need amino acids. How do you make sure you got all the amino acids you need? Say all your essential amino acids. You're a vegetarian. See, for someone who's not a vegetarian, needs meat. There, you got all the things to make meat. Got all the essential amino acids and the non-essential amino acids. The difference, a non-essential amino acid is one your body's like, oh, I don't have enough of that. Take this other amino acid and poof, boom, poof, magically turn it into the one we need. That's a non-essential amino acid. You transmogrify it into something else. An essential amino acid is one you can't replicate, you can't produce. You can't turn some other amino acid into it. Then you have to get it from your food. And like I said, if you're a meat eater, eat some meat. You've got all the things to make meat. If you're a vegetarian, you want to get all your essential amino acids, don't just eat corn or eat beans. Have both of them in the same meal, and then you've got all the essential amino acids. Anyway, so yeah, problems for vegetarians. Moving on, but they're talking iron here. They weren't even talking protein. So sorry for the tangent. Okay, again, we're talking about anemia. Not enough red blood cells are being produced, and another type of anemia you could have there is renal anemia. Renal meaning kidney. This means you're not getting enough EPO produced. If your kidneys are diseased, the kidneys are not producing enough EPO, and then you're not having EPO tell the bone marrow to make the red blood cells mature. How do you fix it? Give people synthetic EPO, fake EPO, or non-natural EPO. Anyway, moving on. You could also have a plastic anemia. This is if the red blood cell, if the bone marrow is being messed up. It could be, somebody has leukemia. You might intentionally destroy their bone marrow with radiation. So, or with chemo. So, what can destroy bone marrow? Drugs, chemicals, radiation, viruses. If it's not something that's being done for a cancer treatment, we may not know why. We just know that the bone marrow is dying. And this won't just affect red blood cells. It'll affect the white blood cells. It'll affect the platelets, the thrombocytes, all the stuff that's being made in the bone marrow, all the formed elements. And that means that you'll not just have anemia, you'll have trouble clotting. Your immune system will have problems because you've got no white blood cells. What do you do to treat it? Well, one thing would be to transfuse new blood in, give people blood. And long-term treatment, you could put transplanted stem cells into the bone marrow to try to restore the bone marrow and give it the ability to cope, the ability to survive. All right, so we've got the All right, so we could also have anemia from too many red blood cells being destroyed. They're being lysed, they're being broken prematurely. If you don't know it by now, you should know that that word lyse or lysis means to break open. So breaking open, we call that hemolytic, again, reference to the word lyse, hemolytic anemia. This could be caused by giving people a transfusion of the wrong kind of blood. The immune system will attack the new blood and break it down. It could also be caused by infections that cause it to be broken down. And the hemoglobin, If you have abnormalities, you have a problem with the way that the hemoglobins you're making, thalassemia or sickle cell anemia, then they're not going to work right. Anyway, so too many red blood cells being destroyed. Let's talk about thalassemia, then we'll do sickle cell. So typically found in people of Mediterranean ancestry. One globin chain is faulty or absent, so you have a messed up shaped hemoglobin. And the red blood cells are thin, they're delicate, they don't have enough functional hemoglobin. There are a lot of subtypes, they have a lot of different varieties. Some are mild thalassemia, some are really extreme. If they're very severe, you might need monthly blood transfusions just to keep you alive. All right, sickle cell anemia. You've got a mutated hemoglobin. It's problems in the beta globulin. Remember, we have two alpha and two beta globulins to make a 4 polypeptide hemoglobin, and it's only 146 amino acids. Each amino acid has one codon that tells you 3 pieces of DNA nucleotide to tell you what amino acid to use. One is wrong, and that's enough that under low oxygen conditions, the hemoglobins are going to start sticking to each other. You're going to form these long crescent shape. Oh, there's a picture of it. And you can see if you take a glutamic acid and replace it with a valine, those hemoglobins start sticking together. You form long curved rods that stretch the cell, sickle it. That only happens under low oxygen conditions. Anyway, so the cells will become crescent shaped under low oxygen. That could be under exercise, especially exercise at high altitude. These red blood cells that have the wrong shape, they can easily break because those long curved rods of hemoglobin will punch through the plasma membrane. They can easily, because their shape, get stuck in capillaries, and this will cause not enough oxygen to be delivered to the tissues and huge amounts of pain from low oxygen conditions. You get low oxygen, you got to build up lactic acid. Running wind sprints and your legs burn. Imagine feeling like that from any kind of exercise. All right. So sickle cell is most common in people of African descent from the areas where there is lots of malaria. People who have sickle cell do not, or even sickling trait, it reduces the chance of getting malaria. But it says malaria can kill 1 million people a year. Individuals with 2 copies of the modified hemoglobin get sickle cell anemia. People with 1 will get sickling trait. It's a milder form, and they have a better chance of surviving malaria, but also a better chance of surviving sickle cell than if they had 2 copies of the gene. So in places with lots of malaria, there is an adaptive advantage to having only 1, to being a heterozygote, having 2 different versions of the gene. One parent gives you the normal version, one gives you the sickling version. If you want to know more about this, I've got a link to the Howard Hughes Medical Institute article on the discovery of the cause of sickle cell. Anyway, moving on. So, how do you treat this? Acute treatments, that means things you got to treat right away. You could inhale nitric oxide. Well, what's that do? That causes the blood vessels to dilate. It makes it harder for the sickled cells to get caught. It gives you better delivery of blood, and that means better delivery of oxygen to the tissues. What can you do to prevent sickling? Hydroxyurea could be given, and that will cause the formation of fetal hemoglobin. Fetal hemoglobin doesn't have the same, it's not exactly the same, and it doesn't sickle as well. You could also do stem cell transplants, gene therapy, and again, nitric oxide for vasodilation. The gene therapy is really interesting. Right now, we have people who are talking about using CRISPR, which is the newest, hottest, coolest form of gene modification. All the scientists love to play with CRISPR to fix it. Remember, this problem is only one nucleotide, and that messes up one amino acid. If they can cut out the mistake and replace it with the correct one, what I'm saying is if they can cut out the valine and put in instructions that say make a glutamic acid, then you've got no problem. Okay? Every bone marrow cell that you fix can make normal hemoglobin from that point on. Let's move on. All right, so you've seen the pretty picture. Polycythemia, you've heard me use that term before. That's the opposite. That's when you got too many red blood cells. The blood is thick, sludgy. It doesn't flow well. People who abuse EPO used to get this problem. People used to abuse blood transfusions. That's another way that athletes used to do blood doping. Their hematocrit could go all the way up to like 80% instead of the normal 45. Blood's super thick. Yeah, it can carry a lot of oxygen, but you can't pump it. You could have heart attacks trying to pump it because it's so thick. How do you treat it? Try to dilute it, or therapeutic phlebotomy, blood draws. You draw some of the blood out. You could also have secondary polycythemia. If you have really low oxygen levels, you could make too much EPO, and then you mature too many red blood cells. All right, so polycythemia, here's where I was talking about the athletes abusing that. In the olden days, before it was like take too much EPO, the way they used to do it, where I said they would abuse blood transfusions, they would take they would draw out their blood, like, oh, I'm giving blood, and they would take the blood out. Then they'd spin it down, take the plasma out, and then before an event, they would put the cells back in. Put a bunch of cells in, the blood's full of cells, full of the ability to carry oxygen. But if you make it too thick, then the heart has trouble pumping it. Remember I said that's when you had athletes who were having heart attacks and dropping dead. You also had a risk if you had an untrained medical personnel, like having a coach do it instead of a trained doctor. Sometimes they put the because we keep blood refrigerated, they put it in when it was too cold, and then you have all kinds of problems from that. Anyway, all right, so we have talked about red blood cells. We're going to talk about white blood cells, leukocytes. Leuko means white, cyto means cell. Yeah, pretty straightforward. You already know, never let monkeys eat bananas. That's the types of white blood cells in order from most common to least common in a healthy individual. So neutrophils, lymphocytes, never let monkeys, monocytes, which will grow up to be macrophages. Monkeys eat eosinophils and B basophils. All right, moving on. So leukocytes are white blood cells. They're the only formed element that has a complete nucleus and organelles and everything you should have in a cell. That means they can live longer. Now, they're only about 1% of the total blood volume. You remember we were talking about the Buffy coat? Or I said about, it's actually less than. You should normally have somewhere between 4,800 and 10,800 white blood cells per microliter of blood. Their main function is to defend against disease. They can leave the capillaries, literally crawl through the walls. We call that process diapedesis. Yes, know that word. They can move through the tissue, through the cells, by acting like an amoeba and oozing around with pseudopodia. Call that amoeboid motion. And often when they're doing that, they're following positive chemotaxis. That's a fancy way of saying they are sniffing their way around, following a trail of chemicals, and they move following the chemicals. That was highly productive. All right, moving on. Okay, so leukocytosis. Okay, leuko for white, cyto for cell. Your white blood cell count, if it's over 11,000 per microliter, then you've got something going on, probably an infection that's causing you to make more white blood cells than normal. The white blood cells can be grouped into two categories. Granulocytes, those have little granules in them, and agranulocytes, the ones that don't. It's pretty easy to tell them apart. Okay, if they have "phil" in the end of their name, like neutrophil, eosinophil, basophil, they are filled with granules. I know that's spelled differently, PHIL, and it means philos for lover, but don't worry about that. Fill means they're filled with granules. Now the eosinophils, those are the red granules that we talked about before, big red granules that make the cytoplasm look really pinkish. Basophils, they're basic loving, so they got the B's to remind you of blue. Don't follow that guy.
No, I ain't.
All right. The basophils are lovers of the blue dye, basic dye, and so they look kind of purplish blue in their cytoplasm. The neutrophils are neutral. They're not basic, they're not acidic, and so they got a little of the blue, a little bit of the pink. But the big thing about knowing the neutrophils is the lumpy partitioned nucleus and the color overall. It has granules, but the neutrophil granules are much smaller, much harder to see. So don't depend on looking at the granules, depend on the overall color of the cytoplasm and the overall level of lumpiness in the nucleus. Now the agranulocytes, they don't have granules. A is a prefix which means without, and agranulocytes don't have granules. In their cytoplasm, examples that would be lymphocytes and monocytes. I told you already, if it has "phil" in the name, it is filled with granules. There is another mnemonic you could use. One of them that I used is the agranulocytes should remind you of someone beautiful, without marks, without any dots on their face, no freckles. That person, think of the Mona Lisa. So you have the monocytes and the lymphocytes. It's just another mnemonic device. It doesn't matter either way. Oh, and wow, here it is finally. The mnemonic device to remember their decreasing abundance in blood. Never let monkeys eat bananas. Neutrophils, lymphocytes, monocytes, eosinophils, basophils. That means basophils are the most rare, neutrophils are the most common. Neutrophils are probably around 70% of the white blood cells that exist in normal conditions. All right, so whenever we do our spinning the blood down in the centrifuge, we have the Buffy coat. And the Buffy coat contains the platelets and the leukocytes. When we look at the leukocytes, we have granulocytes and agranulocytes. The granulocytes are the neutrophils, the eosinophils, basophils. I have another mnemonic device to help you remember that. I still think the best one is filled. The granulocytes are filled with granules. But you could remember that there's a guy named Ben, and he's eating his fill. Okay, So Ben, basophil, eosinophil, neutrophil, whatever works for you. Agranulocytes, you can see the lymphocytes and the monocytes. Again, never let monkeys eat bananas. See that neutrophils are the most common. There's this huge amount here. And the agranulocytes, the most common is the lymphocytes. So never let. You can see the two biggest right there. All right, here are the pretty pictures of them. Now, you remember I said never let monkeys eat bananas, but let's do, they start with the granulocytes, neutrophils, eosinophils, basophils. I would put them the other way, Ben, basophil, eosinophil, neutrophil, but whatever. They're full of granules. You see basophils are full of the basic loving granules. Basophil means basic loving, but that means they love those dark purplish, bluish granules. See the big flecks of dark purple blue in the cytoplasm? The eosinophil, it's got more of the red going on here. I know it's purplish red, but it's got a lot more of the red. You can see big lumps of it everywhere. The neutrophil, it's lighter colored than either one because it's got granules, but they're small. They're not easy to see. The big thing that tells you about the neutrophil, one, they're super common, but two, look how incredibly lumpy the nucleus is. I mean, this one is broken into 3 pieces here. You got only this tiny little thread connecting this part of the nucleus to that part. They don't look at all like the nucleus in your textbook drawing of a normal cell. All right, agranulocytes. I've said this before, but I'll say it again. The lymphocyte is little, and it's the smallest of the white blood cell types. It is almost all nucleus with very little cytoplasm. That cytoplasm is pretty clear, and it's just a little bit bigger than a red blood cell. Now, the monocyte, the M should remind you of massive, because this is the biggest of the white blood cell types. Notice it has a kidney-shaped, kidney bean-shaped nucleus, and this monocyte is what's going to grow up to be a macrophage, the big eater. All right, so, and that's the third most common of those. Yes, you are going to need to be able to identify those on site for the test. I will have pictures and you will need to know what they look like. Moving on. Is that not there? All right, so granulocytes, you got 3 types, the basophils, the eosinophils, the neutrophils. They are larger than the red blood cells. They don't live as long as red blood cells. They are lobed. The nucleus has lobes or lumps, especially the neutrophil. The neutrophil, those lobes are barely connected by these thin threads. They have cytoplasmic granules that are stained. The eosinophils stain more reddish. The basophils stain more bluish purplish. The neutrophils, the individual granules are super small, but they have some of both, so they stain in between the two. All of these are phagocytes, at least a little bit. That means they can go up to something, you'll surround it, eat it, pull it in. All right, so neutrophils are the most common. Remember, never let monkeys eat bananas. Neutrophils are the most common. 50 to 70% of your white blood cells are about twice the size of red blood cells. They are neutral because they like the acid and the base. That means their granules are going to have some of the red granules, some of the blue granules, and they contain hydrolytic enzymes or antimicrobials. The microbial proteins are called defensins. They literally will go up to a bacteria, release these hydrolytic enzymes. What that means is they release enzymes to lyse, to break down the cell. Another way of describing the lumpy nucleus is to use the phrase polymorphonuclears, because the nucleus has lots of shapes, lots of lumpy weirdness. The cell could have as little as 3 or as many as 6 of these lobes that are barely connected by hair-like threads. Again, weird looking nucleus, not what you're going to see in a generic drawing of an animal cell. All right. So the neutrophil is very phagocytic. It likes to eat things that don't belong. Uh, Especially it's good at bacteria. It is also in your white blood cells, kind of your first line shock troop, because you have most of them. Those are the white blood cells that, if there's an injury, they're going to be on site first. They kill microbes through a respiratory burst. The cell will make hydrogen peroxide. Literally, this white blood cell makes its own hydrogen peroxide, stores a little peroxisomes, or bleach, and it then goes up next to a bacteria and says, "Aha! I have a little gift for you," and it kills them, which is pretty badass. Sorry. Anyway, they also could have those proteins called defensins. They can merge with a phagosome, a element that helps phagocytize, to eat or break down. And they're calling them spears. I think it's better to describe them as donuts. But they have, might take the far end. But they punch holes in the membrane. That's the easy way to think of it. They punch holes in the membrane of bacteria, and the bacteria spill their guts out. And yay, we're happy for it. All right, I'm going to pause this right here. And